Hi, my name is Katherine Scangos. I'm a psychiatrist and a neuroscientist with an interest in developing novel therapies for neuropsychiatric conditions. I'm an assistant professor at UCSF in the Department of Psychiatry, and there I conduct quantitative circ-based neuroscience research to identify electrophysiological biomarkers of mood disorders and new forms of responsive brain stimulation. I led a clinical trial of personalized closed-loop deep brain stimulation for treatment-resistant depression, and I also co-direct the TMS, Transcranial Magnetic Stimulation and Neuromodulation Clinic at UCSF. Currently, I'm also a medical director at Neumora Therapeutics, where I work to integrate multimodal biomarker development into clinical trials on a large scale. So, thank you very much for the invitation to present today. As an overview, today I'll talk about the goal of personalization within depression treatment, the use of neurostimulation to specifically modify dysfunctional neural circuit activity, and the clinical trial that we're running at UCSF designed to test the feasibility of personalized closed-loop deep brain stimulation and the initial results of that trial. Here are my disclosures. And here are the learning objectives for today's talk. One is to state 3 approaches for personalized deep brain stimulation, to identify 3 important properties of stimulation response, to understand the concept of closed-loop control, and to understand how spectral and network features can be integrated to identify individual depression subnetworks. So depression is a very common condition. It has a prevalence of about 300 million people worldwide. It's one of the leading causes of disabilities, and while many people can be managed very well with behavioral therapy or pharmacotherapy, there's a cohort of people who are resistant to really all standard therapies that are available right now. And the poor response rate may be due to the etiological and diagnostic heterogeneity in depression. There are a lot of studies now that suggest that depression can be related to alterations in a variety of biological systems shown here on the slide. And so it's really unlikely to be a single disorder. And this, as many know, led the NIH to develop a research framework for investigating mental disorders that integrate across these many levels of information to explore basic dimensions of functioning rather than the disorders themselves. So for example, anhedonia or dysphoria or cognitive dysfunction or problems with arousal or regulatory systems that cross over diagnostic boundaries. Novel technologies such as cortical or subcortical brain stimulation are focused on circuit dysfunction models of depression. And so they're a potential solution for patients that are resistant to all available therapies, particularly because symptom dysfunction can be mapped to dysfunctional circuits and then specifically targeted. So intracranial brain stimulation, such as deep brain stimulation, has shown promise as a treatment for refractory depression. In this treatment, a pulse generator is surgically placed in the chest or in the skull, and it delivers stimulation through implanted electrodes to an area of the brain. So it typically targets a single structure in the brain in all subjects with largely a fixed and constant stimulation dosing. And this approach has been successful in Parkinson's disease and epilepsy. DBS emerged in 2003, led by the pioneering work of Helen Mayberg. And in open-label trials, patients, even with the most severe depression, achieved remission. And so there was really a great deal of excitement because it was one of the first treatments that targeted specific brain circuits. But unfortunately, these results were not consistently replicated across 3 randomized controlled studies. And so the figure here that I'm showing shows a trial of ventral capsule stimulation where efficacy was assessed at the 16-week time point. And the lack of difference here between the active and the control conditions, it may be due to the trial design, which is some people suggest that the 16-week time point may be in retrospect too early. However, inter-individual response heterogeneity also likely played a role. And so novel strategies in intracranial brain treatment stimulation are needed rather than abandoning the treatment altogether. So personalization of therapy has been proposed as a means to improve the outcome of medicine in general, and it's been shown to be able to do that. The variable presentation of symptoms across individuals with depression is evidence that different neural circuits may underlie different depression biotypes, and it speaks to the need for personalization within depression as well. And in this image from Leanne Williams' work, she highlights specific circuits underlying specific depression symptoms and suggests that knowing the neural circuits involved for each patient might inform our clinical decisions for that patient. And we argue that this type of personalization is also needed in brain stimulation as well. And in fact, multiple anatomical stimulation targets have been identified and shown to have promise in small studies, and these are shown here on the left, and include areas such as the subgenual cingulate and the ventral capsule, which are common structures, but now other regions as well, regions within the reward circuitry, the OFC, for example, as well. And the fact that stimulation across these multiple sites can improve symptoms in different sets of people suggests that personalization in brain stimulation is possible. So I'll talk briefly about the two most common DBS targets. The majority of work has been done in the subgenual cingulate by Helen Mayberg, building on the cortical limbic dysregulation hypothesis of depression. And she proposes that you can think of depression as the net effect of failed network regulation, which can be modulated from different nodes. And she identified the subgenual cingulate as a key node that addressed what she perceived to be one of the primary symptoms of depression, dysphoria. And she also found that there was increased blood flow in the subgenual cingulate in both induced sadness in healthy subjects and in depressed subjects, and that the overactivity decreased as depression improved, or naturally or with successful treatment with antidepressants or with deep brain stimulation. And she's gone on to optimize the targeting in the subgenual cingulate using DTI. So she identifies a particular region of the subgenual cingulate based on patient's own anatomy that leads to the best outcome. And you can see here, it's the intersection between these three different white matter tracts. The second target that's been most commonly utilized is the ventral capsule region. This is part of the reward pathway and targets anhedonia, which is another key symptom of depression. And it was at first used as a region for OCD with capsulotomy and then DBS. And it was found to reduce depression symptoms in these patients as well, and then was used as a primary target for depression itself. And Samir Sheth's group is another group that's focusing on personalization within DBS, but they're using a different strategy. They're implanting DBS leads in the ventral capsule and subgenual cingulate, as well as temporary electrodes in additional brain regions to explore the large stimulation space, the parameter space, so varying stimulation intensity, frequency, pulse with polarity, and using current steering to look at the volume of tissue activation to find the ideal parameters for each individual patient. So in deep brain stimulation, traditionally electrodes only deliver stimulation, but they can't sense neural activity. So there's really no way to read out the effect that the stimulation has on the circuit you're targeting. But it's also possible to implant microelectrodes, arrays, or leads that can continuously record activity or sense it. And that's what's being shown here. There's a patient with spinal cord injury who has an array planted in M1 that senses neural activity. And when one particular pattern is observed, it can move this experimental arm. And later on, a patient's own arm itself. So you can see from this, a personalization of therapy includes not only where you stimulate, but also when you stimulate. And with motor disorders, you have visual feedback. You can see that the arm is moving or the tremor of Parkinson's disease is reduced. But in depression, you can't necessarily visualize an emotion or a thought. You have to rely on subjects or physicians' reports of symptom severity state at each moment in time, but we can apply the same principles. So with the addition of neural sensing, a patient's own physiological activity can selectively trigger stimulation when a pathologic state is detected. And then you can read out the effect that the stimulation has on the circuit that you're targeting. And so that's the idea behind closed-loop deep brain stimulation. Stimulation is delivered only when symptom severity is high, which may reduce side effects, prevent adaptation, and ultimately improve efficacy of this type of treatment. So there are a couple of devices that have the capability to continuously sense neural activity. So what I'm showing here is the FDA-approved and commercially available NeuroPace RNS system, which is the one we were using in our study. But the biggest challenge for closed-loop deep brain stimulation in psychiatric disorders is finding a symptom-specific neural biomarker that can trigger stimulation. And a biomarker like this has not previously been found for depression. But there are ways to do this. So direct neural recordings and focal electrical stimulation have been used in the pre-surgical evaluation of patients with epilepsy in order to identify each individual's seizure focus and map out important functions that need to be spared. So this is language or motor control. And these electrodes are typically left in place for about a week while patients remain in the hospital. And we thought that a mapping stage like this could also enable personalization for psychiatric disorders. And we now have trials at UCSF for depression, pain, OCD. And the idea here is to extensively map cortical limbic circuitry at the individual level and find a biomarker of high symptom severity state that could be implemented then into closed loop treatment with one of these chronic devices. So the ideas behind this goal were developed through years of research through a DARPA SUBNETS program led by Eddie Chang at UCSF. And in this research, motion circuitry was mapped out in patients with epilepsy to identify novel stimulation targets. Patients with epilepsy have a high rate of depression and anxiety. And then biomarkers of anxiety and depression were also investigated. And building on this initial work, we designed a clinical trial to test the potential efficacy and safety of personalized closed-loop neurostimulation for major depression. The goal here was to perform the mapping stage in patients with primary depression and then implement closed-loop neurostimulation with the NeuroPace RNS system. We named the trial the PRESIDIO trial. And so in the first stage, we implant 10 temporary 16 contact electrodes to map out personalized stimulation and sensing targets. And then we begin to identify a neural biomarker of each patient's symptoms. We remove those electrodes. In the second stage, the biomarker is integrated into the closed loop therapy with the NeuroPace system. And then finally, in the third stage, we perform a randomized controlled study of the initial efficacy and feasibility. Our study population focuses on patients with treatment-resistant depression. The major inclusion criteria to note that are different than previous trials are, one, we require evidence that neural circuitry can be modulated as evidenced by a response to ECT, but not a sustained response. So patients may respond after one or two treatments, but they need to keep getting ECT to have the benefit. And then two, that we require that there's sufficient variability in self-reported symptoms that and this is a requirement because we need to identify the biomarker, and this helps us do that. So here's the overall study trial design. As I mentioned, the first stage of the trial is the implantation of the 10 electrodes across 10 different brain locations within the cortical limbic circuitry. The patient stays 10 days in the hospital or laboratory setting and we conduct stimulus response mapping to identify the brain target where stimulation best improves depression symptom severity. And then we also pair resting state neural activity with clinical depression scales to identify spectral biomarkers of a high symptom burden. So if we were able to find a brain site where stimulation leads to a consistent change in clinical symptoms, then participants meet criteria to move on to the second stage. So here I am showing the results of the 10-day clinical mapping study for our first patient. So during this time, we performed bipolar stimulation survey across the electrodes in the ventral capsule, the subgenual cingulate, hippocampus, amygdala, and the orbital frontal cortex. And what we saw is really a diversity of very specific responses across different stimulation sites and stimulation parameters. And we just delivered 90 seconds of stimulation, and we could see a really broad array of responses. So some examples of this included tingles of pleasure with 100 hertz ventral capsule stimulation, neutral alertness. I feel less cotton and cobwebs, she said, with 100 hertz stimulation in the subgenual cingulate. She experienced a calm pleasure. She described it like reading a good book with 1 hertz low frequency stimulation in the OFC. And it was really astonishing to see these effects. And through this experience over the course of the 10 days, we really learned the roadmap to her brain to the point where we could predict the resulting emotion or mood that would develop prior to the stimulation itself. And I think it was an incredible experience for the patient as well, who had really been stuck in one state for years and years, and to suddenly have stimulation that changes your mood and to suddenly feel relief was just extremely life-changing for her. So after we did the stimulus response mapping, which is just brief and the patient knows what we're doing, we wanted to further explore the brain sites and the stimulation parameters that elicit the positive response with sham-controlled stimulation. So here we assessed the effective stimulation using a couple of different clinical measures. We used a 5-point Likert scale combining subjective responses with physician-rated affect. We used visual analog scales of depression, anxiety, energy, and we used a 6-question subscale of the HAM-D17 to assess the short-term changes in depression severity. So results from these scales across our different trials suggested that stimulation in this patient largely modified 2 dimensions, the arousal dimension and the mood valence dimension. And we found that there were 3 different stimulation paradigms in the patient that all reliably improved mood, but were associated with different effects on the level of arousal or activation. And that's what's shown here. You can see in this rectangle here, you can see arousal in dark blue and mood valence in blue here, and then somatic symptoms in pink or peachy orange. And then in the second row of the heat map, you can see the arousal was pink and the mood valence was blue. So, you know, while both of these stimulation conditions improved mood, they had different effects on the symptom of arousal, the arousal dimension. So 2 of the paradigms, 100 hertz stimulation of subgenual cingulate and ventral capsule, were consistent with prior DBS studies. And the third was a novel location and stimulation condition, low frequency stimulation applied broadly across the orbital frontal cortex. And the responses to stimulation at these sites were consistent across trials. Ventral capsule and subgenual cingulate were consistently activating, while the OFC was consistently sedating. However, the effect they had on mood was state dependent. And that's what you can see in these triangle plots here. For example, in the OFC, shown in the first one, the effect of stimulation was positive and calming if we delivered the stimulation during a high neutral arousal state. But it worsened mood if we delivered the stimulation during a low arousal state, causing the patient to feel excessively drowsy. And the opposite pattern was observed for the subgenual cingulate and the ventral capsule regions where stimulation increased arousal. So we found that the effects of the stimulation were state dependent. And while this patient's primary symptom was anhedonia, because it was anhedonia, she perceived the most consistent benefit from stimulation in one region of the ventral capsule. Again, one area of the reward circuitry, which made sense. So we also found that the response to stimulation was dose dependent, as shown here for the ventral capsule stimulation, on both mood in red and, in gray, and arousal, shown in red. And we found that the response to stimulation was sustained beyond the stimulation period itself, and in some cases even up to 40 minutes for the ventral capsule. And so together, these results, the dose dependence, the persistence, and the state dependence together suggested to us that closed-loop stimulation may be a feasible treatment option that was worth further exploration and may even be necessary. So in addition to the clinical mapping, we also did biomarker exploration during this period. So we had the patient perform 335 VAS scales of depression and anxiety over the 10 days, which we used to define two symptom severity states, a low and a high symptom severity state. And we identified the 15 minutes of resting state IEG activity that coincided with the clinical measures where one of these two states was present. And then we identified the spectral activity features that discriminated the high and low symptom states using two different cross-validated supervised machine learning models at different spatial resolutions. In the first model, which is shown on the top here, spectral power was averaged across all contacts within a brain region, so it was a region-level model. And here we found that gamma power in bilateral amygdala, right OFC, left subgenual cingulate, and right hippocampus had high state discriminative potential. In our second model, the feature set was defined by spectral power for each electrode contact, so including across all the brain regions, and that allowed us to account for interregional variability due to the electrode positioning. And with this model, we found bilateral amygdala gamma power alone was actually sufficient for detecting the high symptom severity state. So it was simpler than we initially anticipated. So it suggests that amygdala gamma power is a biomarker of the highly depressed state in this individual. So we found the ventral capsule was the brain site where stimulation led to consistent improvement in symptoms, and amygdala was the site of the biomarker activity. We next examined the connectivity across the cortical limbic network to determine whether the ventral capsule and amygdala nodes constituted a structurally and functionally connected subnetwork, so that ventral capsule stimulation directly influenced amygdala biomarker activity. And to us, this provided further justification for selection of these sites for the placement of the sensing and stimulation leads. So to examine the effective network connectivity, we used evoked potential mapping, EP mapping. So we delivered single pulses in one region and looked at the evoked potential in a distant contact. And then we identified the N1 and N2 peak deflection magnitude using standard approaches. And so that's what I'm showing here in this first circle plot in A. It's the directed graph for the N1. And the percent circumference here indicates the strength of the connection between any two of the brain regions. And the color here indicates the starting location. So for the ventral capsule, you can see the yellow connections pointing towards the hippocampus in black. Connectivity strength and the direction was then quantified by calculating the weighted in degree. The weighted in degree is the weighted sum of significant connections pointing towards a node, and we calculated the out degree, which is the weighted sum of connections pointing away from the node. And we did this in order to identify the most influential hubs across this network. And interestingly, we found that the ventral capsule had the highest weighted out degree, suggesting it has strong connections to other brain regions, and the amygdala and hippocampus had the highest weighted in degree, suggesting they receive connections from many other brain regions. And this is consistent with what we might want for a stimulation and sensing selection and sites for deep brain stimulation. So we then examined the axonal fiber tracts that might mediate this effective connectivity using DTI, and this was aided by one of our collaborators in neuroradiology, Leo Sugru, and we found that there was also dense structural connectivity between the right ventral capsule and amygdala, and it was comprised of two fiber tracts, the stria terminalis, which terminates in the bed nucleus, and then the ansa peduncularis. And then finally, we found that stimulation at the contact that led to the best improvement was 2 + 3-. When we stimulated there in the ventral capsule, we saw improvement on 11 of the 14 trials. And when we looked at the change in gamma power on the trials where we had both pre and post VAS scales and resting state EEG activity, we found that there was a reduction in amygdala gamma on both trials where symptom severity improved, and that reduction was absent on trials where symptom severity worsened due to an alternate stimulation paradigm, or if it remained steady due to minimal symptoms at baseline. So together, we characterize this complex interplay between symptoms, mood state, and neural stimulation. We demonstrated the importance of a patient's symptom profile in interpreting the clinical response to stimulation. And together, these results suggest that the time a patient spends in any particular mood state could be a consideration in the selection of a stimulation target. It also suggests that a biomarker can be identified and that we can identify a limbic subnetwork that both reflects depression severity and can be modulated to decrease clinical symptoms. And this figure here just shows our approach, identifying a stimulation target, identifying the biomarker, and then looking at the connection between those two sites in 3 different ways, looking at functional connectivity and structural connectivity, and then the direct effect of the stimulation. So in stage 2 of the trial, we implanted the NeuroPace device. This is after we removed those temporary electrodes, the patient recovered. Then we went back and we implanted the NeuroPace device with 2 separate sensing and stimulation depth leads based on the mapping study. And we used the DTI that I just showed you in planning of this surgery so that the contacts interface with those 2 fiber tracts, the stria terminalis and the ansa peduncularis. And then we planned it out that way, and then we were able to successfully replicate the positioning of the contacts we were looking for. We then assessed whether we could also replicate the clinical effect of the ventral capsule stimulation that we saw in the mapping study. And we found that at the expected contacts based on anatomy, we again saw acute symptom improvement across the majority of trials. And the patient described it as pleasurable and energizing, just as in stage 1. And in fact, we saw the opposite effect with stimulation in the amygdala, which was a control. And you can see that stimulating there made her symptoms generally worse. So furthermore, in closed loop stimulation, in addition to the frequency and pulse width and current amplitude, we also have to consider the burst duration of stimulation as well. So to do this, we tested the clinical effects of intermittent stimulation at different stimulation burst durations over a 15 and 30-minute period. And we found that just 6 seconds of intermittent stimulation at 1 milliamp was clinically effective. And something that was important to us, it was below the patient's perceptual threshold, meaning that she couldn't detect these on-off state transitions. Which we didn't want because we didn't want her to go through her day feeling the stimulation go on and off. We also found that we were able to replicate the symptom-specific biomarker for the closed-loop control. So we used the NeuroPace system, telemetry wand to stream ECOG in the lab. And then we recorded 10-minute trials of continuous amygdala activity while the patient engaged in naturalistic activities. This might include looking at her social media, talking to family on the phone, watching clips of movies, or even attending her virtual therapy appointment in the lab while we recorded her neural activity. We then paired the neural activity with the clinical severity ratings, and we again found that amygdala gamma power was highly correlated with both the VASD, the Visual Analog of Depression Scale, and the HAM-D6, and it significantly differentiated this high symptom severity state from the low symptom severity state, just as we found in the first stage. We then implemented the closed loop therapy, so we turned her stimulation on at home such that 6 seconds of stimulation would be delivered every time this biomarker was detected. And we also found that the number of detections within a 10-minute period was highly correlated with the visual analog scale of depression, the VASD, and the HAM-D6 as well. And so that's what you see in this final graph. You see the number of gamma detections is correlated with the HAM-D6 as well as the gamma power. So over 2 months, she went home, she received the closed loop therapy, and we analyzed that data. We found that on average, there are 468 detections per day with minimal stimulation at night. Stimulation did interfere with her sleep if we gave it late at night, so we set a stimulation cap of 300 therapies per day. And this meant it usually stopped around 5 p.m. And so that's equivalent to just 30 minutes of total stimulation per day. This is way less than standard DBS and about equivalent to a TMS treatment. And we found that with this amount of stimulation alone, we saw a rapid improvement in symptom severity as measured by the VASD and the HAM-D6, and depression as measured by the MADRS. And of course, we need a randomized controlled trial to determine efficacy, so we can't say very much about it here, but we can say observationally that this subject's MADRS score dropped from a 33 the night before stimulation was turned on to a 14 at the first measurement 12 days later. And then it dropped below 10 remission within several months. And she remains in remission today. So it's been almost 2 years at this point that when she first had her first surgery. So to further then test whether the closed loop algorithm triggered stimulation in a manner linked to the patient's symptoms and not randomly, we examined then the relationship between a morning symptom severity rating that she did at home and the daily detection count over the 2-month period. And we used dynamic time warping so we could nonlinearly align this daily symptom severity and biomarker detection count time traces. And you can see here how wide they are. This is the purple and the gray lines. We found that the fluctuation in daily symptoms was significantly associated with the fluctuation in number of biomarker events detected by the device. And this suggests that it detects change in symptom severity better than chance rather than just stimulating intermittently. So in summary, we replicated the effects of the mapping study with the NeuroPace device. The effects of the clinical response to the ventral capsule stimulation was positive and energizing, and amygdala gamma power was again found to be predictive of the symptom severity state. And as I mentioned, this patient's depression is currently in remission. She's doing really well. She started driving again after not driving for a lot of years. She's taking computer programming classes, Python classes, and engaged in going to school with a goal of moving back into the workforce. So it's really been amazing to watch this transition in her life. Okay, so, all right, so we have a lot of work left to do. We don't yet know how variable these findings will be across individuals. We have implanted a couple of additional subjects at this point in different brain locations, so we don't think the same biomarker and treatment site is going to be the same across all our patients. And we also don't know whether the short-term response that we're seeing will lead to a long-term response in major depression. So we need to do more work, but the results are really encouraging. And it's important to note that this framework is not unique to depression. It could be applied to a broad range of neuropsychiatric conditions, as long as they involve network dysfunction. So the understanding of circuit activity related to pathologic states and the dose-effect relationship of circuit modulation could also help provide domain knowledge to aid development of other novel treatments for depression. Many times these relationships are studied in animal models, but in psychiatric disorders, the relationship between circuit function and disease phenotype is difficult to assess without asking how it's doing. So we think that developing these human circuit biomarkers in small patient numbers may allow for back translation to animal models for more extensive testing of these circuits and stimulation responses within which neural populations are involved, and that this information could then be forward translated again into larger patient populations. And the unprecedented access to the brain really allows us to do this. So the work I presented today is made possible due to a broad, collaborative, multidisciplinary team that includes Edward Chang, who conceptualized much of the work and led the DARPA Subnets project, Phil Starr, Ankit Kambadi, Kusama Kul, and Andrew Crystal. Andrew leads the broader brain stimulation program in the psychiatry department. And of course, our other collaborators, Bikram Rao in neurology and Leo Sugru in neuroradiology, and our funders as well. So thank you very much.